So Christian, I think we can turn the YouTube live on. Hello, everybody. So let's start the WHO information meeting on the composition of influenza vaccines for use in the Northern Hemisphere 2024-2025 influenza season. Dr. Mike Ryan, the executive director of the WHO's emergency program, couldn't make it in person today, but he did a video recording his welcome and the opening remarks. Now let's run the video.
Dear colleagues, greetings and welcome to the WHO information meeting on the composition of influenza virus vaccines for use in 2024 to 2025 Northern Hemisphere influenza season. I first would like to thank all of you from both the public and the private sectors for your tireless efforts in supporting influenza preparedness and response. I have opened many of these biannual meetings on influenza vaccine composition. Each one reminds me of how critical the work is to the WHO Global Influenza Surveillance and Response System. Since its establishment in 1952, GISRIS has grown to now comprise 160 institutions in 129 member states, with around 50 additional member states affiliated informally. Each year, seasonal influenza kills up to 650,000 people, and the threat of a pandemic caused by an influenza virus is real, as signaled by continued sporadic infections of human and avian and swine influenza viruses. Over the past several years, there has been a growing geographic spread of avian influenza AH5 virus, with spillover to mammals. GISRIS has been the bedrock of influenza preparedness and response, thanks to the enormous support of hosting countries and international partners, including all of you. It cannot go unnoticed that all of your work has deep roots in global health history. We just celebrated 70 years of GISRIS, and now we are looking ahead to the next 30 years. Beyond influenza surveillance, preparedness and response, GISRIS has been leveraged for other respiratory viruses, including RSV, since 2015, and SARS-CoV-2 since March 2020. We are advancing GISRIS so that expanded GISRIS, or e-GISRIS, plays as important a role for other respiratory viruses as it has for influenza. As GISRIS provides sentinel surveillance to monitoring the co-circulation of respiratory viruses, the assessment of relative risks associated with different viruses is instrumental to countries' decision-making. In relation to the topic of today's meeting on influenza vaccines, the GISRIS practice of operating year-round presents an excellent model for other viruses and diseases of epidemic and pandemic potential. The influenza vaccine selection has provided strong inspiration for how WHO makes recommendations for COVID-19 vaccine composition. Currently, the co-chairs of the WHO TAG COVAC are also directors of two GISRIS WHO collaborating centres. Public-private partnership based on collaboration via PIP and GISRIS are a model for addressing similar challenges of other viruses and diseases. As seen during the COVID-19 pandemic response, industry is an essential player with new influenza vaccines in the pipeline. We look forward to furthering joint efforts to constantly improve influenza prevention and control. The GISRIS network and its partners, including you, are a vital asset to global health. I thank you for your dedication and support. I wish you another successful round of the influenza vaccine cycle for the Northern Hemisphere in 2024-25. Thank you.
Mike said, I hope you can hear me. At this moment on, we're entering into a new cycle, the new vaccine cycle for the Northern Hemisphere 2024-2025 at full speed. I would like to also add my welcome to everybody to the venue here in Montreux, Switzerland. I would also like to welcome colleagues connected via YouTube Live at the moment. So welcome everybody to the meeting. My name is Wenqing Zhang, head of the Global Influenza Program in WHO headquarters, Geneva. Without further ado, let's move on to the next agenda item is the selection of the chair of this WHO information meeting. So as in the past, the chair of this information is always be one of the WHO's temporary advisors who attended the meeting and made the recommendations that will be announced. This time we have invited Dr. Paul Bamford from TGA, the Therapeutic Goods Administration Australian, to be the chair of this information meeting. Let me ask the floor if there's any objection or any concerns against Dr. Paul Bamford to be the chair of this information meeting. I hearing none. So without further ado, I just pass over to Dr. Paul Bamford to run the meeting.
Thank you very much, Wen-Ching. It is my great pleasure today to be the invited chair. So thank you very much for having me today at this meeting at the composition of the influenza virus vaccines for use in the 2024-2025 Northern Hemisphere influenza season. Many of you know me. I'm Pearl Bamford, as Wen-Ching has already mentioned, and I'm here from the Therapeutics Good Administration as an essential regulatory laboratory in Australia. I hope you have all been able to enjoy some of the beautiful views that we're going to be seeing today. And if you have not, maybe have a look at the balcony and look at the wonderful snow-capped mountains during coffee break. I won't delay things further. I would like to also open up the microphones to have self-introductions of the other advisors at this consultation meeting. So we might start at the end of the table in Kanta.
Thank you, Pearl. Good morning, everyone. I'm Kanta Subbarao, the director of the WHO Collaborating Centre in Melbourne, Australia.
Good morning. My name is Otto Engelhardt. I'm from the WHO ERL at the MHRA in the UK.
Good morning. I'm Rebecca Condor, the interim director of the WHO Collaborating Centre at CDC Atlanta.
Good morning, Richard Webby from the WHO Collaborating Centre in Memphis, US.
Good morning. This is Hideki Hasegawa, WHOCC at Tokyo, Japan.
Good morning, this is Diane Wang from WHOCC Beijing, China.
Good morning, everyone. My name is Nicola Lewis, and I'm the director of the WHOCC in London.
Good morning. I'm Vasily Marchenko from WHOCC in Koltsovo, Russian Federation.
Good morning. It's Dr. Ziping Yi, a representative from WHO ARL Food and Drug Administration in the USA.
Thank you. And thank you all for coming in person today and for those that are streaming online. So for us, just a bit of a background, the advisors came here and we started the consultation meeting on Monday. We've been busy reviewing the genetic and antigenic data, the epidemiological, serological, and modelling data. It's been an extensive week going through a lot of information, but we have made some decisions about the recommendations of the composition of the next influenza vaccines for the Northern Hemisphere. And without further ado, we will be going through the data packages, starting with the H1s, the H3s, the Bs, and then moving on to zoonotic. So I will be holding questions until after all the data packages for seasonal viruses are presented. And so please hold your questions until that point. So without further ado, I will be asking the first presenter, Professor Nicola Lewis, to welcome to the podium and present the H1N1 data. Thank you.
Good morning, everybody. It's a great pleasure to firstly present to you the influenza activity summary, and then I will move on to presenting the information on the AH1 PDM09 viruses. So here you can see the number of specimens that have been processed by GISRIS by week. In blue, you see the number of specimens we had in 2021, and in the slightly darker blue, you see the specimens from 22, and in the yellow, you see the specimens from 23 through into 2024, shown in red. And when we take a look at the global circulation of influenza viruses since 2017, you can see that we have these epidemic peaks. And here you also see that the PDM09 viruses are shown in the lighter blue color, the AH3 viruses are shown in the slightly more turquoise color, and then the subtyped viruses, the unsubtyped viruses that are A subtype are shown in the darker green. When we look at the period from 2023 to 2024, you can see that in 2023, we had this very large peak and a bimodal distribution to our seasonal influenza viruses. And through into 2024, you can see that we've had the uptake in the viruses, a predominance of influenza A viruses, but with some activity with the B Victoria lineage viruses too. If we zoom in a little bit to look at the circulation of influenza viruses by hemisphere, on the right-hand side panel, you can see that the Southern Hemisphere continued to have activity through September, October, November, and indeed into the early 2024. Whereas we see in the Northern Hemisphere, really activity picked up from November onwards. When we look at the countries and areas and territories that have shared specimens and viruses with the WHO collaborating centres, you can see that this is really quite a markedly large geographic distribution, with many countries in many regions sharing specimens with the WHO CCs. When we look at influenza activity and the global distribution by type and subtype, you can see that we have heterogeneity in the kinds of types and subtypes that are circulating in different geographic regions. And we also have this tail to the Southern Hemisphere season where activity was still maintained towards the early part of 2024. So you can see in the Southern Hemisphere, really the influenza B viruses continue to circulate, whereas when we look at the distribution for more Northern Hemisphere regions, you can see that there's a predominance of influenza A viruses. When we look at the viruses that have been genetically characterized by the WHO collaborating centres, you can see here in this distribution that certainly for the September 2023 through to January 2024 period, we have had reasonably equal proportions of AH1 PDM09 viruses and AH3N2 viruses characterized by the WHO CCs, but indeed an increase in the number of B Victoria viruses that have been genetically characterized too. How this breaks down in terms of the total number of viruses that have been antigenically characterized in the past 3 seasons, well, again, this figure shows you that we've had an increase in the number of viruses that we've been antigenically characterizing as well as genetically characterizing in the past season. Moving on to the AH1N1 PDM09 viruses. Again, this is a very similar plot to that which we've just previously seen for the overall activity. And you can see that the yellow line represents the AH1N1 PDM09 virus activity through the end of 2023, when we saw an uptick in influenza activity for the H1 subtype from about week 40 to 43 onwards. And this has also tailed off in the early 2024 period. So really from week 1 of 2024, we have seen a reduction in activity of AH1N1 PDM09 viruses detected by GISRIS. Again, when we look at where the activity has been located for the AH1N1 PDM09, we see again heterogeneity in terms of the activity by regional distribution. So in some countries, certainly in the Afro region and the EMRO region, we have seen high levels of activity, whereas perhaps we've seen a reduction in activity in more eastern regions. Here we can look at the activity for AH1N1 PDM09 viruses that were detected from the 1st of February through to the 31st of August based on HA sequence availability. And you can see in the right-hand and the left-hand side of this plot, the distribution of genetic subclades and the diversity that we had through this reporting period. So we had a fairly large amount of genetic diversity in the AH1N1 PDM09s through this reporting period. If I now show you the activity and the clade distribution for the most recent reporting period, for the time through from the 1st of September to the present, you can see that we've had a reduction in genetic diversity and really a predominance of the 6B1A5A2A and the 6B1A5A2A1 subclades circulating. You can also see from this plot that the distribution and the proportions of these two subclades, so the 5A2A and the 5A2A1 subclades, differs by country and by region, with some regions showing a proportion that's increase in 5A2A1 activity, whereas other regions have a predominance of the 5A2A viruses. When we look at the viruses that have been characterized antigenically for the AH1N1 PDM09 viruses, you can see that all the collaborating centers have been characterizing a number of these viruses. And also these viruses have been characterized in this reporting period in both collaborating centers that are located in the Northern Hemisphere and also in the Southern Hemisphere. This is a phylogenetic tree showing the evolution of the AH1N1 PDM09 viruses in this reporting period. And as I've already mentioned from the clade distribution figures that we've just seen, we see the 5A2A and the 5A2A1 viruses continuing to circulate. And if you look at the tick marks in this these bar plots to the right-hand side of the phylogenetic tree, you'll see these are colored by the region in which these viruses have been detected. So, for example, these viruses in the 5A2A1 group here that are colored in green have been most recently detected in Europe. But we've also seen the 5A2A viruses detected in nearly equal proportions in the European region. And I'd like to thank my colleagues at the University of Cambridge, Derek Smith and Sarah James, for producing the figures we are now going to view. So we can also analyze the genetic information and look at the frequency of the different subclades that are circulating. And this is some analysis that we have had a look at in collaboration with our colleagues at NextStrain. And you can see here on the left-hand side, we have the frequency trajectories of the 5A2A and the 5A2A1. The 5A2A is shown in this orange color and the 5A2A1 is shown in the green color. And you can see that the frequency trajectories vary by region. As an exemplar of some of the analysis we look at, we could also take some of the motifs that we see, the particular substitutions that we see in the genetic information, and we can look at the frequencies within that sequence information by region as well. And this is shown on the right-hand side of the plot. I've just taken an example here of the amino acid substitution K142R, and you can see that we can actually track the different frequencies that we see in the data by geographic region too. And we can do this for a number of different substitutions and different parameters that we have in the sequence data. We also phenotypically analyze these viruses, and we use the HI hemagglutination inhibition assay to analyze the antigenic properties of the H1 PDM09 viruses. And then we use a technique known as antigenic cartography to quantify the antigenic relationships amongst these viruses. And again, I'm very grateful to my colleagues at the University of Cambridge for producing these antigenic maps. And what these antigenic maps show you is the interrelationship between different viruses that are circulating in the human population. The viruses that are shown in gray here are represented by gray circles, and they're viruses that are older, so they've not been circulating recently. The colored viruses are the viruses that have been circulating in the current reporting period. And you can see that we've colored them by the particular subclade of AH1N1 PDM09 that we've been looking at. So if you look at the key at the bottom, the 5A2A viruses are shown in this maroon color, and the 5A2A1 viruses are shown in purple. And you can see for both the collaborating center data from Atlanta and also from London that the 5A2A1 viruses are very antigenically similar to each other, but they're also very antigenically similar to the 5A2A viruses because they co-locate in the antigenic maps. If we interpolate a serum circle over this, what the serum circle shows us is the number of viruses that fall within an eightfold of the homologous titer for a particular serum that we're looking at. So here you can see that the viruses are highlighted, the sera that are highlighted here are the sera that are anti-sera that are raised against the A Victoria 4897 2022 egg or the A Wisconsin 67 2022 cell. And you can see within the collaborating center data from Atlanta that all the viruses that have been antigenically characterized in this reporting period, whether they're from the 5A2A or the 5A2A1 subclade, are within an 8-fold of that particular referent antiserum. This pattern is very similar in the data from the collaborating center from London. We can also look at the number of viruses that are out with an 8-fold, and you can see here that for all collaborating centers, these viruses were well recognized by antisera raised against the A Wisconsin 67 2022 like viruses and or the A Victoria 4897 2022 like egg viruses. We also look at human post-vaccination serum. And what we're doing here is using this as a surrogate to understand how human sera are recognizing these viruses and whether there's been the potential for antigenic drift through this reporting period. And you can see in these data that have been kindly shared by the WHO Collaborating Center in Tokyo, that the panels of antisera that we have used to analyze these currently circulating strains of AH1-PDM09 viruses are well recognized by the antiserum panels that we have used. Now on to the antiviral summary. When we look at neuraminidase inhibitors for this reporting period, of the 2,089 clinical specimens and isolates that were examined for neuraminidase inhibitor susceptibility, either by genetic or by phenotypic analysis, 22 viruses showed evidence of reduced susceptibility. And of these, 22 that were phenotypically tested, 9 showed reduced or highly reduced inhibition by NAIs. When we look at the endonuclease inhibitors, of the 1,656 PDM09 viruses examined either by genetic or phenotypic analysis, 1 had a substitution at Y24H in the viral polymerase and showed reduced susceptibility to the endonuclease inhibitor baloxavir marbazil in the phenotypic assays. So in summary, the influenza A H1N1 PDM09 viruses, phylogenetically, AH1N1 PDM09 viruses circulated globally and predominated in most geographic regions. The hemagglutinin genes of the viruses that were genetically characterized belong to the 6B1A5A2 clade. And as we've already seen, this 5A2 clade is split into 2. The subclade 5A2A viruses were detected in Europe, Oceania, the Middle East, Africa, Southeast Asia, and Central America. The subclade 5A2A1 viruses within the group, represented by A Victoria 4897 2022, predominated in the US, the Caribbean, Japan, and Europe, whereas viruses within this subclade, represented by A Wisconsin 67 2022, predominated in Brazil, Europe, and Southeast Asia. When we summarize our phenotypic analysis, post-infection ferret antisera raised against the Northern Hemisphere 2023-2024 and the Southern Hemisphere 2024 AH1N1 PDM09 vaccine components. So this is the cell culture propagated A Wisconsin 67 2022 and the egg propagated A Victoria 4897 2022 from the 5A2A1 subclade recognized the 5A2A and the 5A2A1 virus as well. When we look at the post-vaccination geometric mean titers, these were not significantly reduced for the most recent circulating AH1 PDM09 viruses when we compare them to titers against the cell culture propagated A Wisconsin 67 2022, PDM09-like, or the egg propagated A Victoria 4897 2022 like viruses. Thank you.
Thank you very much, Nicola. And we'll be now moving on to the presentation of the H3 viruses. And I'd like to welcome Dr. Hideki Hasegawa to the podium. Thank you.
Okay, good morning. This is my pleasure to present the discussion about the AH3 and 2 viruses. First, this is the trend of the detection of the H3N2 viruses by GISRS. The most recent trend is shown in the color in yellow of the 2023, and it increased towards the winter season of the Northern Hemisphere and peaked at week 49 and then decreased. And continuously in 2024, the number detected by GISRS is decreasing. This map shows the global activity of H3N2. And as you see here, the H3N2 virus is predominated in Asian area. And in Europe and in Africa, the similar proportion of the virus between the H1 and PDM09 and the H3N2 was reported. And in Oceania region, towards the end of this reporting period, the number is increasing. This is the antigenic tree made by our colleague in the University of Cambridge. The vast majority of the viruses detected this reporting period fall into belong to the 2A3A1. This is further diversified into the subclades, but provisionally, this is designated as H.1 to H.4, but it may change because of the confusion of the hemagglutinin H and this subgrade H. So it may change, but provisionally it is H.1 to H.4. And the color suggesting the region where the virus is isolated. This map showing the distribution by the subclade. As you see here, the 2A3A1 viruses colored in green. And specifically, the H.1 subclade is light green, and H.2 subclade is colored in darker green. As you see here, the vast majority of the viruses belong to either H.1 or H.2 subclades here. And the H.2 predominated globally, and the H.1 virus predominated in South America and Africa and Japan. Okay, this is also the trend of the diversity of the by subclade in each region. As you see here, that the color is the same as the previous ones. The light green suggesting the H.1 and the darker green is H.2. The vast majority of the virus towards the end of this reporting period, it belonged to the either H.1 or H.2 in most of the regions shown here. And this is the number of the viruses characterized antigenically by the WHOCCs. As you see, the number is increasing in the NID and the viral and the CNIC. That's here. Okay, this is the antigenic analysis, the result of antigenic analysis. The virus was characterized that the reactivity against the anticellular against the parent anticellular against the cell culture propagated A Darwin 6 2021 like cell or the egg propagated A Darwin 9 2021 like viruses. And so here, the 44% of the this against Darwin 6 or 48% Darwin 9 fall into the lower reactor. On the other hand, the same assay by HI assays, the anticellular against the parent anticellular against the Massachusetts 18 2022 like cell virus or the egg-grown A Thailand 8 2022 like virus was shown here, and the nearly more than the 90% or nearly 90% of the viruses characterized that reacted well against these viruses, anticellular against by these viruses. This is the example of HI assay of H3N2 virus done by the Biddle, the research in Melbourne. And you see the almost the vast majority of the viruses belong to the 2A3A1 reacted very well against cell-grown or egg-grown Thailand 8 viruses shown here. But with the exception of some reduced reactivity in several some viruses, which is belong to the provisional subclase H.2. Okay, this is antigenic cartography made by a colleague in University of Cambridge. As you see here, this is the antigenic map of the 2A3A1 viruses. And these viruses clusters very well. This is the result of the hemagglutination inhibition assay done by CC Melbourne, or the focus reduction assay done by the Biddle. And if we put the serum cycle of the Thailand 822 antisera, the vast majority of the viruses is located within the cycle, with the exception of some viruses in the peripheral here, which is a provisional subclade H.2. This is a summary of the serology data from all collaborating center and ELL. And that in this serology test, we used the pediatric or adult on the elderly serums who were received the previous Northern Hemisphere vaccine containing the 2A vaccine. So compared to the reactivity against the 2A viruses, several panels showing the significant reduction against the 2A3A1, especially to the H.2 subclade shown here. Okay, in summary, in some countries, areas, and territories reporting influenza A viruses, H3N2 subtype predominated. The significant H3 activity was observed in Southern Africa and Asia. An HA phylogenetic HA of circulating H3N2 viruses in this period fell into the single clade 2A3A1. There is a further diversification of 2A3A1 into several subclades. Provisionally, it is called designated as H.1 to H.4. And H.2 have predominated in most regions, with H.1 predominating in Africa, Southern America, and Japan. And virus expressing CRE2 HG gene, including the subclades, were ungenetically closely related and ungenetically distinct from the 2A.1 viruses. And ferritin cell raised against the A. Darwin 6, 2021, which belonged to the 2A clade. HA clade 2 viruses from multiple subclades were genetically well recognized, with some subclade recognized less well. And virus expressing clade 2B HA showed a reduction to 2A3A1 viruses. And the ferritin cell raised against A. Thailand 8, 2022, which is the clade 2A3A1. HA clade 2A3A1 viruses were well recognized, but showed reduced recognition to some provisional H.2 viruses. And human serology studies with the serum panels from the individuals vaccinated with the Darwin 6, 2021, like 2A viruses, some recent H3N2 viruses from the H.1 and H.2 genetic subclades were significantly reduced in some serum panels. Antiviral susceptibility, there are 3,046 H3N2 viruses collected and analyzed since the 1st September 2023. None showed genetic or phenotypic evidence of the reduced inhibition to neuraminidase. Of 1,450 H3N2 viruses collected and analyzed since the 1st September 2023, 4 showed the genetic or phenotypic evidence of reduction, reduced susceptibility to Barack-Severe Marburg seal. Thank you.
Thank you very much, Hideki. And it is my great pleasure now to introduce Dr. Rebecca Condor to present the B viruses.
Good morning, everybody. So now I'm going to discuss the characterization of influenza B viruses. So during the reporting period of September 2023 through January of 2024, we can see here over the last 4 years of circulation, the influenza B viruses have significantly circulated in years 2023 through current period of 2024. We can see in 2023 that there was a large increase in detections of influenza B during weeks 5 through 18, and then continual detection and increase again in 2023 and since week 40, and a large increase leading up to where we are now in 2024. So a lot of influenza B detections by GISRIS network. This view takes a look back since 2019 and looks at the influenza detections of influenza B, type B, that then have individual analysis for their lineage determination. We can see epidemics due to influenza B throughout this time period with differing levels of detections in the network. And in the color orange, you'll see the influenza B viruses that only have the type information. But in the turquoise and the blue, we have the B lineage information. So the blue line represents the B Yamagata lineage, and it's very difficult to see, but this blue line actually follows closely along the x-axis and shows very little detection of B Yamagata since 2019 by the GISRIS National Influenza Centers. However, we can see significant peaks of activity of the B Victoria lineage detection since 2019, viewed in the turquoise detection line here. So we can see significant epidemics due to B Victoria viruses through the GISRIS network since 2019. This map shows the influenza B activity from September 2023 till now, with various shades of yellow. Although the majority of the map is quite pale in yellow, influenza B co-circulated with influenza A during this time period. Where we're seeing more significant increases of activity include parts of countries in Central America and South America, countries in Africa, and a large number of countries in East and Southeast Asia. This pie chart shows the amount of viruses with lineage information available. Now, depending on the surveillance source, sometimes we only get a type result for influenza B, but our national influenza centers and our collaborating centers perform the B lineage assay. And in this case, 52% of the influenza B viruses during this time period were typed as B Victoria lineage, and 0 were B Yamagata. So now we'll discuss the virus characterization for B Victoria lineage viruses. This large phylogenetic tree, again, provided by University of Cambridge, shows the evolution of the B Victoria hemagglutinin gene over the last several years. There was a significant genetic event in 2020 that led to the triple deletion of 3 amino acids in the hemagglutinin of the B Victoria gene. This was both a genetic and an antigenic drift variant. This particular virus has continued to circulate since 2020 and continues to have different genetic diversity and has split into several subclades. This includes the V1A3s, so all of those are the triple deletions. They accumulated additional changes in the hemagglutinin to become the 3A viruses. And then these viruses further diversified into 3A1 and 3A2. And over the last couple of years, there's been co-circulation and regional differences in which of the 3A1 and the 3A2 subclades were detected. 3A1 viruses were predominantly found in East Asia and China. However, 3A2 viruses since 2022 have had a global pattern of circulation and detection. If you recall back to the large amount of activity in the 2023 beginning of the year of influenza B, we can see that this was all responsible due to the viruses belonging to the 3A2 clade, with large detections in Europe, Central and South America, Oceania, and North America and Asia. As we moved through the middle of 2023, you continue to see the 3A2s in the Southern Hemisphere. And as we go into the Northern Hemisphere season, since September 2023, you again can see several detections of the 3A2 viruses in Asia, North America, and parts of Europe. So this is a very large scale view. And now we'll look more at the amount of genetic diversity that's been occurring in the hemagglutinin in the 3A2 viruses themselves. So 3A2 viruses share three changes in the hemagglutinin at position 127, 144, and 203. And that you can see at the base of the tree. The other subclade that I mentioned earlier is the 3A1, and this shares changes at position 220 and 241. Very few viruses from 3A1 were detected during this time period, and no viruses from the former 3 clade were detected. In the hemagglutinin, for the last reporting period, the majority of viruses from the 3A2 subclade had the additional substitution in the hemagglutinin at position 197. During this reported period, there was co-circulation of subgroups within the 3A2s with the additional mutations on top of the D197E substitution. This includes a change of position 129. And here you can see the global distribution. Really all regions that saw 3A2 saw viruses from this subgroup. And then we had 2 evolutionary events with the same change circulate, and that's a change of position 183 from E to K. Some of these viruses had an additional change of position 128. And you can see several viruses in recently detected in Asia from this particular subgroup. And at the top, viruses with the E183K, and you can see circulation in parts of Asia and North America, with some also having another change at position 128, but in E to a K. So we're continuing to detect the 3A2 viruses, and we're continuing to see additional diversity in the hemagglutinin. So now that we have the genetic information, we'll then integrate that with our antigenic information. But first, just a map to show the detection and sequencing results from our national influenza centers and WHO collaborating centers. Again, showing how widespread influenza B viruses have been detected in the last 2 reporting periods, and that they have belonged vastly to the 3A2 HA clade. In summarizing the total number of viruses antigenically characterized by CCs, for this reporting period in red, all collaborating centers had data available. And as shown in the activity map, China had a lot of influenza B viruses, and so had a lot of antigenic characterized viruses available. An overall summary with our FAIR anisera using hemagglutinin inhibition assays to look for reactivity. Here we can see that FAIR anisera raised to the vaccine viruses, the B Austria 135, 9417, 2021 like, either cell or egg propagated viruses, showed good cross-reactivity across all WHO collaborating centers, to the viruses available for testing. So greater than 99% reacted within less than eightfold in the HI compared to the homologous titer of the reference viruses. This shows a recent HI test from our WHO Collaborating Center in Beijing. And here's where we can see the antigenic differences between the triple deletions. Those from clade 3, 3A1, and 3A2 in the reference panel. You can see significant reductions against the recent 3A2 viruses with these ferret antisera raised to these former clades. However, the reference panel against recent 3A2 viruses, including the vaccine viruses, show good reactivity against the viruses circulating from 3A2. As shown in previous presentations, we take the cumulative data over time of the antigenic information and try to understand in antigenic space where the genetic viruses, the genetic variation that we're seeing in the hemagglutinin clusters. So we have two different colors on this particular map. In yellow, we can see the former reference viruses for the clade 3 viruses that were the previous vaccine viruses. And then different shades of green, we have the 3A1 viruses shown in data from CDC China, down here in this quadrant. And then up in the left here and over here on this side, we see two slight different colors of green, one lighter and one a little bit darker. And these represent the 3A2 viruses from this reporting period. Those a little bit darker include viruses with the additional substitution at 197, which represents the majority of viruses tested. With these maps, we can see that 3A2 viruses continue to be antigenically very similar to each other. If we now look at the sera circles for our reference viruses, we can see good coverage of 3A2 genetic diversity by these ferret antisera. And where viruses are outside of the clade 3A2, we see reduction. So it's always important to then take this information that we gather from the ferrets and look at post-vaccination human sera. So a subset of the genetic diversity that I described in the hemagglutinin, viruses were then put into human serology post-vaccination analysis. These show results from Northern Hemisphere vaccinees from the US and Japan, spanning age groups from 6 months to greater than 65 years. In all panels of sera and across the diversity of the viruses from the 3A2 tested, we saw no reductions, significant reductions in the GMTs compared to the cell B Austria reference virus for the vaccine. And this landscape analysis shows our non-inferiority analysis across all collaborating centers and includes human sera panels from China, Japan, US, and the UK vaccinated with the 2023-2024 Northern Hemisphere formulations. We can see very few of the panels or viruses tested had potential for inferiority. So in summary, for antivirals, we tested over 1,600 B Victoria viruses by either genotypic or phenotypic analysis, and 4 showed evidence of reduced or highly reduced inhibition by NAIs. We also looked at the PA gene for endonuclease inhibitor susceptibility, and no viruses showed evidence of reduced susceptibility to baloxavir. Okay, now to summarize the data for B Yamagata lineage viruses. There have been no confirmed detections of circulating B Yamagata 1688 lineage viruses after March 2020. So now I'll summarize influenza B. Only influenza B Victoria lineage viruses were available for analysis. In the phylogenetic space, I've shown that 2 subclades circulated. However, the vast majority belong to 3A2. Almost all 3A2s also have that additional substitution in the hemagglutinin at position 197. And I've shown that there's been further genetic diversity within the hemagglutinin of these viruses. Our antigenic characterization using post-infection fair antisera raised against the B. Austria-like reference viruses for the vaccine viruses well recognize the genetic diversity that we've seen in the 3A2 viruses, but again, continues to show that antigenic differences with the 3A1 clade viruses. Results from our human serology studies showed that viruses from 3A2 were well inhibited by human post-vaccination sera. And for antiviral susceptibility, small numbers of viruses with potentially reduced or highly reduced susceptibility to neuraminidase inhibitors were identified, and no viruses showed reduced susceptibility to endonuclease inhibitors. Thank you.
Thank you very much, Becky. Now, that presentation now concludes the seasonal presentations. I will open up the floor for any questions at this point. Okay, I'm hearing none. I'm seeing none. Okay, so now, as you know, this committee not only reviews the seasonal data and of viruses circulating, but we also look at zoonotic viruses that are being detected around the world. And I would like to now welcome Dr. Richard Webby to the podium to present the data on zoonotic influenza. Thank you.
Great, thanks, Pearl, and good morning all again. So I just want to first by acknowledging the contributions of both the GISRIS laboratories, but also particularly our colleagues from the OFLU network and other institutions around the world that have really contributed a lot of data that has gone into the summary that I'm about to present. So if we sort of take a look at what's happened since we met last time in September, you can see the number of zoonotic influenza cases has continued to mount. We'll go through these in a little bit more detail in showing slides, but as a summary, we have seen the variant viruses, so viruses from swine spilling over into the human population, and then also a continuation of viruses from the avian reservoirs infecting humans. All of these infections here, one-off infections, so animal to human infection, but no subsequent transmission. And so let's look at these individually now. If we start with the H5 viruses and data provided to us by our OFLU colleagues, if we look at the dots here, particularly the blue dots, these are reported outbreaks that are reported to the agricultural organizations. If you look just in terms of geographical spread first, so dots across many countries of the world, perhaps not showing up particularly well on here, but certainly some movements of this virus southward, so into the Falkland Islands and now into some of the Antarctica regions of the world as well. So some concern as this virus is perhaps moving down and what it may do if it gets to that continent. In terms of the clades of viruses circulating, the 2344Bs remain the dominant clade of H5 viruses circulating globally. And so most of these light blue dots you can see here are the 2344B activity, with some residual 2321A activity in India and Bangladesh, and the 2321Cs detected in Southeast Asia, particularly in Vietnam, Laos PDR, and Cambodia, as we will hear next with some of the zoonotic cases. So looking at those in a little more detail, so where these viruses have spilled over into humans, there have been 8 cases in Cambodia. All of these are due to viruses with a hemagglutinin of 2321C clade. The severity here ranged from fatal through to very mild disease or with poultry exposure. And again, as you can see here with the age of the individuals infected, a spread of age, maybe with a little bit of leaning towards younger individuals. Similarly to human infections in China, these are with the 2344B viruses in China. A lot of these 2344Bs are paired with the N6 neuraminidase. In birds and many other parts of the world, it's paired with the N1. The 2 cases, again, very severe, the fatal disease, poultry exposed, adults infected. If you're looking at the cumulative numbers here, we are reaching up on 1,000 cases, but these numbers, of course, continue to climb. Antigenically and genetically, although there's some difference in these viruses, and even within Cambodia, some genetic differences in the viruses that caused the February 2023 infections to those that caused the later. All of these are still, luckily at this stage, well covered by our existing stockpile of candidate vaccine viruses. So no new candidate vaccine viruses for the H5s. H9N2, so again, a virus we've seen a lot of zoonotic infections with in this period, 4 additional cases within China. Again, all poultry exposed individuals, so bird to human infections. In this HI table, taken from the Collaborating Center in Beijing, we can see some viruses here that are representative of H9N viruses detected in the recent past, with some of the candidate vaccine viruses here, the Anhui as an example. And these viruses still, in general, react pretty well with ferret antisera produced to that. So again, the H9s, although they continue to accumulate genetic diversity and genetic amino acid substitutions in HA, still pretty well covered by existing reagents. The new virus this time, so this is the first time that this particular virus subtype has been detected in humans. There was a report of an H10N5 infection, again in China. This particular individual had some underlying conditions. It was a fatal case, but had some fairly extensive exposure to the infected bird in this case. If we look at, again, a phylogenetic tree here, produced by the Collaborating Center in Beijing, and we have seen at least H10 influenza viruses in humans before, paired with the N3 and the N8. So the N3 up here, the N8 here in the orange color. These particular viruses belong to the Eurasian genetic lineage of HA. This new infection, the H10N5, belongs to a different genetic group. This particular virus here doesn't have any H9N2 genetic segments, unlike the early human infections. Very much a wild bird virus. It's unclear of the exact extent of the distribution of that virus in more of the domesticated bird populations. So again, this is a little bit of watch to see what happens, but it does appear to be a somewhat isolated case of a wild bird virus infection. Moving on, so to the other source animal population we see providing viruses to humans is swine in this case. So if we look globally, and this is just H1 swine influenza viruses, again with a map provided by our off-fluke colleagues. There is a alphanumeric soup, of course, of clade nomenclature for these viruses. We do see reports or detections of these viruses across many countries. Don't be particularly fooled by the lack of colors in many of the other regions of the world. These viruses are endemic in most countries where there is swine production. So lots of viruses circulating in the animal populations. We've seen a number of instances in this period where these viruses have infected humans with zoonotic infections. Three of them, the H1N1 variants with a couple of different genetic clades. So from both South America, Brazil, and Europe, to an H1N2, infection in the UK. Most cases, these people reported exposure to swine, but not all of them. So we do have some holes in our arsenal of candidate vaccine viruses to some of these variant viruses and viruses circulating in swine. These particular viruses, so the 18332, so again, these are representative viruses circulating in Brazil, Spain, and Switzerland. These are viruses from the 2009 pandemic lineage that have spilled back into swine and continue to evolve there. Just a couple of recent examples here by the Collaborating Center in London, some viruses from swine and variant virus in Denmark. I think see by the lack of reactivity of these viruses to older pandemic seasonal reference viruses has made us make a recommendation to produce a virus, and this is a virus from the recent variant virus from Spain. Again, to cover this group of viruses that we repeatedly see popping up in variant cases. And similarly, with this time the 1B111, so the H1N2 infection, these viruses are viruses that circulate predominantly in the United Kingdom, and were responsible for the human variant case in this period. And again, data here from the WHO Collaborating Centre in London, showing that very, very closely related viruses to the variant case that circulated in swine, again, have really no reactivity to the panel of old human reference viruses, seasonal reference viruses, and similarly, we don't actually have candidate vaccine viruses on the zoonotic side to cover this group. So our recommendation is to make one to fill this hole, and so an A England 2023 like CVV will be developed. So I think that's what I got. Thank you.
Thank you, Richard. Any questions on zoonotic?
Yeah, so thanks, Steve. Yeah, that's an interesting virus. So there has been I guess a genetically, when I say genetically distinct, at least genetically distinct from the H7N9 that I think we're used to seeing in humans. So this is a virus that has been reported in a number of bird species across South Africa, with maybe some spill into neighboring countries now. No evidence yet of human infections with that virus, but there was data presented both from our Offaly colleagues on that virus. Again, it does seem that perhaps some of our older H7 candidate vaccine viruses may produce antibodies that will cross-react with that virus, but it's something that we're watching closely.
Any further questions from the floor? Okay, well, we'll move on to the next agenda item, and I would like to welcome Wen-Ching back to the podium to announce the recommendation for the composition of influenza vaccines for use in the upcoming Northern Hemisphere 2024-2025 season. Wen-Ching, over to you.
Thanks very much, Paul. So I'm going to present the conclusion of this WHO consultation just finished yesterday. So as always, I would like to start with acknowledging the WHO's Global Influenza Surveillance and Response System, GISRS, which currently comprises 160 institutions in 132 countries, areas, and territories. It, of course, includes also 151 national influenza centers in the country. Twice a year, WHO recommends the composition of influenza vaccines. This work is all based on the year-round surveillance of this global system. Taking this opportunity, I would also like to acknowledge not only the GISRS, but also the countries, the national authorities in countries hosting these GISRS institutions. And also would like to acknowledge those institutions which are need to be, or national influenza centers to be, these institutions as well, which already started contributing to the GISRS system, although they don't have this official recognition or designation yet. I would also like to thank partners, GISRS partners, different agencies, and friends of GISRS. Without this collaborative work or collective efforts, it would not have been possible for GISRS coming through 72 years now to today's scale of operation and work. So from Monday to yesterday, Thursday, WHO convened a consultation on the composition of influenza vaccines for use in the upcoming Northern Hemisphere season. This is the first time all in-person meeting since COVID-19 pandemic. This consultation was chaired by Dr. Diane Wang, Director of the WHO Collaborating Center in Beijing, China, and co-chaired by Dr. Kanta Suparao. Also, I would like to take this opportunity to give a very special thank to Kanta Suparao, because probably as some of you know that Kanta is leaving the WHOCC region in Melbourne in May. And I would like to thank Kanta for her essential role in the past seven years, indeed, in the GISRA system, as well as her significant contribution to the pandemic, the COVID-19 pandemic response. In the consultation, 10 WHO temporary advisors who are directors of WHO collaborating centers and essential regulatory laboratories of GISRAS attended this consultation. At the start of the meeting, the interest declared by these advisors were disclosed to all meeting participants. In addition, this consultation was also attended by 32 observers from GISRAS, national, regional public health agencies, academia. We also have colleagues joined from World Organization for Animal Health, FAO, and OPLU Network. In addition, colleagues and staff from WHO headquarters and regional offices also attend the meeting. The consultation analyzed massive volume of data generated and or collected by the global system GISRAS and also from other sources. And this is a long list of the data that has been looked into. The committee of the WHO's temporary advisors, based on the analysis, they evaluated the potential options of the composition for the upcoming season and concluded the recommendation. So it is recommended that for the upcoming 2024-2025 Northern Hemisphere influenza season, trivalent vaccines contain the following, and this is a list of the recommended vaccine composition. As you can see that this list is the same as the recommendation for the Southern Hemisphere 2024 developed or announced in September 2023. But if compared with the recommendation for the Northern Hemisphere 2023-2024, the past Northern Hemisphere, there's one update on H3N2 component. So further to the WHO's September 2023 recommendation, it remains the opinion of the WHO's Influenza Vaccine Composition Advisory Committee that the B. Yamagata lineage antigen should be excluded from influenza vaccines as it is no longer warranted. National or regional authorities should make decisions regarding the transition to trivalent influenza vaccines in their jurisdictions. Where quadrivalent vaccines are still used, the fourth component, the B. Yamagata lineage component, remains unchanged from previous recommendations, and that is a B. Phuket, B. Yamagata lineage-like virus. So the technical reports supporting this recommendation, I think is already already live on the WHO's website, or it will be alive very soon. There are a couple of documents that provides detailed information, including the WHO's technical report for the seasonal FAQ, as well as a report of summary of the of the characterization of the zoonotic influenza viruses. In addition, the candidate vaccine virus as well as the reagents for this round of vaccine cycle are also available or already published on the WHO's website. To point out that for pandemic preparedness purposes, WHO has been updating the candidate vaccine viruses for zoonotic influenza, also published on the WHO's website for pandemic preparedness purposes. I think this is all from my side. I hand back to the chair.
Thank you very much, Wen-Ting. Now, with these recommendations now being announced, we obviously now need to move on to the candidate vaccine viruses available for manufacture and those potency reference reagents that you'll be then subsequently using. And I'd like to invite to the floor Dr. Ziping Ying to present these.
Okay. I'm going to present two important components. One is for vaccine production. Another one is to standardize of the vaccine antigen in the final product. First part is the candidate vaccine viruses that can be directly used for propagation of the virus in embryonic eggs or the cells, and then later on processed as a final antigens. However, the antigen has to be standardized to make sure what amount of antigen in the final product. So candidate vaccine viruses can be directly used in propagation in eggs or embryonic eggs or in the cells, and later on processed as the final antigens. Or the genetic information can be used for production of recombinant protein or mRNA. So this slide shows the candidate vaccine virus for egg-based production. The recommended virus is the Victoria 4897 2022. There are some viruses is antigenically similar to the Victoria 4897. Usually the wild type viruses may not grow so well in eggs or in cells. So there some labs produce the reassortant, which is to get the surface antigen from recommended virus and internal genes from high growth virus, so that viruses can grow well to produce the vaccine antigens. So this is panel shows the viruses. I wouldn't go into read all those viruses. And then this part shows the reassortant. Then what kind of method do they use for reassortant, or and also where you can get this from? I think as when Cheng mentioned, those information will be in WHO website, so you can get updated information from there too. And I have to mention that there is the accession numbers, and pay attention to that one because you should use this information to get sequence information for your recombinant protein production. And here shows the recommended viruses for cell-based vaccines. Again, this the viruses and the reassortant and where you can get this from. As Wenqing mentioned, because the recommendation for Northern Hemisphere is exactly the same as Southern Hemisphere recommended viruses 6 months ago. So we can still continue to use those reagents for production for Northern Hemisphere. And I'm not going to read the whole thing here because it may be too small for you to read. And I think that the information can be found in WHO website. And this slide shows the again the viruses for cell-based vaccine production. And again, is the viruses that can be used for egg-based for the bee viruses. And this shows the viruses for cell-based vaccine production. And as mentioned, Wenqing just mentioned that we still have this bee Yamagata bee bouquet. We still provide those information for those who wanted to, for the transition from quadrivalent to trivalent or for the research purpose. Okay, here's down to the potency reagents. Because the vaccine antigen have to be standardized. The current method that we use potency reagents, which compose two parts. One is the antigen itself, and also the antibody that use the purified HA to immunize animal and to get the polyclonal antibody. And those reagents can be used to standardize the vaccines, protein-based vaccines, including recombinant protein. And this slide shows the potency reagents for H1N1 egg-based. So here shows where this the viruses being produced as reference antigen, and this is the viruses they use this the reassortant, and this part shows the lot of each of the reference antigens, and this part shows the amount of the antigen that you use a specific lot, and this is the readout. And here it shows where you can get this, where you can get this material from. And this antiserum is produced in sheep, and it's, as I said, the sheep was immunized with a purified HA or recombinant protein, depending upon what kind of material you prepared. And again, this shows the cell-based, the potency for cell-based vaccine. Again, this shows the reference antigen, and this shows reference antiserum. And again, this information you can use to acquire over those material from the these agencies. And this slide shows the potency reagents for H3N2 because as I said, this is this material has been used in Southern Hemisphere, so you can continue use those information for production or for the incoming Northern Hemisphere stream production. And this slide shows the cell-based use for cell-based antigen standardization, and this shows the egg-based for B Australia 1359417 2021 like virus. Here it goes to the FUCAT. As I said, this material is still available for those who want to use those information or use the material for the translation from a quadrivalent to trivalent. And then this shows cell-based, potency reagents for cell-based or recombinant protein-based vaccines. Again, I think the Winchin already shows this slide, shows this link so that you can get those information on those material from corresponding labs or the agencies. Because after this meeting, we have a following up meeting, those reagents they'll be updated because the reason we have a variety of the reference antigens or reference antibodies or candidate viruses. So the lab is still producing those candidate viruses, and so that they have more varieties of the viruses, and so that people can evaluate which one is will be most suitable in their manufacturing facilities. Over.
Thank you very much, Ziping. Now, I'd like to open up the floor for any questions in relation to the morning session, just before we break for coffee break. No questions? Excellent. So, oh, Steve.
Model the possibility of diverging to a Yamagata-like species, in particular the 149 G2R and the 203 K-to-N mutations, because we've observed this before in the early 1970s. We know where the B Victoria lies antigenically historically, and so rather than just look at antigenic diversity, if we see direction towards a splitting strain, towards a serologically distinct virus, in other words, multiple clades, this would be very useful because it would give us an early warning signal that we might go back to a quadrivalent vaccine or not. It's just a recommendation, mine. Thanks.
Very good point. Thank you very much, Steve. Any further comments from the floor?
Stephen Morgan from Sun. I think my question's a lot less technical than Steve's. Dr. Yu, you mentioned that the CDBs and the reagents, etc., for the Phuket are still available. So obviously manufacturers are transitioning, and as mentioned, you pointed out that countries and regions are also moving with their transition plans from QIV to TIV. And I just wondered, I picked up on the word still available, and I wondered if you'd had a discussion on the duration of availability of the the reagent and assays into materials that would be available to manufacturers?
I think it depends upon the, I cannot speak that for individual agencies. But I think so for now, we have what we have, and it also depend upon demand. If we really need it, we can produce it. I cannot speak that for them, but I think this can be considered from my point of view.
Maybe I can add, so from our perspective as an ERL, the inactivated reagents and antiserum will be available in the foreseeable future, but we have taken all the live CVV viruses off our website. You can't order them anymore. I mean, there may be special considerations, but generally not.
Okay, thank you very much. Any other questions? Comments? I think we've made very good time. We've got 5 extra minutes for coffee break. So I'd like to ask everyone to return back here at 11 o'clock. So you've got 35 minutes. Coffee will be served in the room across the hall. And for those that might not have found it, the toilets are also located just to the left of this room down the set of stairs. So there you go. Thank you very much.